with 4 children. She lives in excruciating pain every single day because she has something called sickle cell disease. It causes her blood cells to be irregularly shaped and block her blood vessels. Not just that, sickle cell disease can cause anemia, organ damage, strokes, and even early death, according to the Innovative Genomics Institute. That was the reality for Victoria Gray, the first ever patient of a CRISPR therapy called Casgevy. She participated in a 2019 clinical trial in Nashville, Tennessee. Now, sickle cell disease is caused by a genetic defect in bone marrow, which causes the production of a protein leading to the creation of half-moon-shaped blood cells rather than round ones. And as according to a NPR article by Rob Stein covering the monumental new treatment, doctors injected Gray's own bone marrow cells back into her after they used the CRISPR-Cas9 gene editing tool to edit the cells so that they produce a protein that would compensate for the defective one. And that's the start of today's talk here, gene editing. So first let's get some background on what exactly goes on in gene editing. According to a paper published in Trends in Biotechnology, there are 3 main gene editing tools. You have zinc finger nucleases, ZFNs. You have transcription activator-like effector nucleases, or TALENs. And you have clustered regularly interspaced short palindromic repeats, or CRISPR, the most recent of the 3. All 3 of them function by creating double-strand breaks in DNA, and they're often called molecular scissors. Now, the paper outlines therapeutic benefits for all 3 tools. However, the older tools, the ZFNs and the TALENs, they have some issues in that they're a lot more time-consuming and difficult to engineer. So the winner of the 3, CRISPR, is actually far more reliable and it's far easier to use. It's far more precise. It's based on an immune defense found in bacteria, which involved incorporating palindromic snippets of viral DNA into its own. So it's essentially an RNA guide paired with a protein that can cut DNA. And this allows people to add or remove any sequence from any location in the genome. And that is a very important technology. So it sounds really cool, right? Now I can help treat Down syndrome, I can help treat sickle cell disease. Maybe we can even reduce obesity among humans. Maybe we can determine the hair color of the next generation. Maybe we can even increase their athletic capabilities. Whoa, let's slow down there before we slide all the way down the slippery slope. As cool as an innovation gene editing sounds, there are some glaring problems. And perhaps the scariest concern is that gene editing tools like CRISPR can be used for a form of new wave eugenics. Now, eugenics is not a new idea. As noted by Daniel Kelps in his History of Eugenics, it started in 1920s America. And it was a way to target the feeble-minded or the undesirable people through non-consensual sterilizations and other hurtful measures. And then later, it was adopted, most famously, by Adolf Hitler and Nazi Germany, used to justify the industrialized mass killings of Jews who were considered a threat to the master Aryan race. Now, imagine that the scrutiny is not on your outer characteristics like skin color, ethnicity, and so forth, but on the very molecules that separate you from the next human being. Gene editing doesn't just let humans treat diseases. In theory, a new despot could control the health conditions and appearances of the people they have power over. Imagine that instead of sterilizing you, somebody has the power to change the very fabric of your being just because they didn't like the way you look or because you don't satisfy their standards of the perfect person. And that's exactly the same nightmare that scientist Jennifer Doudna, one of the pioneers of CRISPR, had, literally. In a biography about her called The Codebreaker, she tells Walter Isaacson how she once had a nightmare in which a pig-headed Adolf Hitler confronted her about the consequences of the gene editing tool she just introduced to the rest of the world. So the bottom line, gene editing can be used to increase authoritarian control, especially if it's combined with the data-hungry artificial intelligence of today. And not to mention there are some other issues as well. For example, accessibility. Right now, a CRISPR treatment costs over $1 million per patient, as mentioned in the codebreaker. Now, if this cost is not improved, that could also alienate CRISPR's access to only the rich. Now, there's also government rules and regulations to look at. And as of now, when it comes to gene editing, they're all a big work in progress, varying from region to region. However, there was general apprehension around allowing germline editing, or allowing gene editing that can be inherited by the next generation. Because sometimes gene edits can cause unintended consequences, especially if you consider the fact that genes have several functions. So if you remove a gene, you can end up damaging far more than you thought. And that made the germline the red line for gene editing. That is until a Chinese scientist, without the authorization of the Chinese government, decided to use CRISPR to create twins without the genes that made them more susceptible to HIV that were initially in their parents. And since the initial red line was crossed, and legislation was already lagging behind innovation, it was pretty unclear what the legal consequences of this should be. So now you might be thinking at this point in the talk, well, the case for gene editing is pretty bleak, right? It seems more like a bioweapon rather than a treatment, but it doesn't have to be. A Google search for the definition of eugenics defines it as the study of arranging reproduction within a human population to increase the occurrence of heritable characteristics considered desirable. That has long been debunked as pseudoscience. At the very base level, eugenics just means to better the human race. But the way these ideas have been executed is often in a very twisted or harmful way. With gene editing, we can redefine eugenics to be a way to alleviate unnecessary suffering that was previously untreatable. After all, it's unethical to not use a solution to a problem if you have it. Now, you might be wondering, well, okay, what happens? How do we stop CRISPR and gene editing from leading to the wake of designer babies and discrimination on the basis of genetic characteristics? Well, the answer to that is to stop rapid commercialization for cosmetic purposes, as happened with plastic surgery. Susanna Haga mentions in her A Brief Comparative History Analysis, Plastic Surgery and Gene Editing, that plastic surgery initially started out as a way to allow soldiers affected whose faces were disfigured by chemical weaponry in World War I to lead more normal lives. But later it became a way to enforce unhealthy beauty standards. Now, plastic surgery is used for some real medical issues, such as for correcting facial or skull structural issues. But as Hagas notes, as the perception of a normal physique began to narrow, consumer demand to fit the norm went up and caused the rapid commercialization of cosmetic plastic surgery before we fully understood its effects. So thus, if we learn from the evolution of plastic surgery, we can avoid making the same mistakes in access and ethics with gene editing. Now let's circle back to the story we started with, Victoria Gray. She was interviewed by Rob Stein 7 years after the trial, and she has reported no relapses of sickle cell disease since. While gene editing does have the potential to usher in a new wave of oppression, it is unjust to ban it altogether when you consider that there are people like Victoria Gray out there, people suffering from painful diseases that otherwise have no treatment. In the end, gene editing is just a tool. And whether its legacy is that of discrimination and oppression, or is of uplifting others, is up to us, the human race, and our intentions when we decide to use it. Thank you very much.